Welcome. So in this video we will learn about the alkaline phosphatase test. Now alkaline phosphatase is an enzyme which is present in the raw milk, and when the milk is pasteurized, this enzyme, that is alkaline phosphatase, get inactivated, and alkaline phosphatase test is used to check the efficiency of pasteurization, whether the pasteurization has been taken place properly or not, but that can be determined using alkaline phosphatase test. And in this video we will learn all about the alkaline phosphatase test in detail. Now alkaline phosphatase enzyme number is here 3.1.3.1. now this naming is as per the International Union of Biochemistry and Molecular Biology Committee, that they have given the nomenclature for enzyme classification, and as per their classification, alkaline phosphatase number is designated as 3.1.3.1. now this is the enzyme which split most phosphomonoesters in the pH range of 9 to 10.5. so alkaline pH is optimum pH for this enzyme, and as the name indicates, alkaline phosphatase. So there is another phosphatase is there, which is acid phosphatase, that is present in milk, and its pH is acidic pH, obviously. So in this enzyme split mostly the phosphomonoester, that we'll see in the slide, that in the pH range of 9 to 10.5. Now alkaline phosphatase has been studied in milk extensively because of its use in the milk pasteurization test. This is mostly used because this is the routine test which is being carried out in dairy industry to check the efficiency of pasteurization. Now the activity of the enzyme is stimulated by some elements like calcium, manganese, zinc, cobalt and magnesium, and these enzymes are under the category of hydrolysis. So there are so many classes of enzymes are there, but hydrolysis are those enzymes which cleave with the chemical bond by hydrolysis. So in this for this enzyme, the monoesterous bond are being broken to corresponding alcohol and phosphate. Okay. Now if you see the significance of alkaline phosphatase, it is written that this enzyme has been used as an indicator of pasteurization. We know that alkaline phosphatase destruction temperature is slightly higher as compared to the temperature of destruction of the indicator organism for pasteurization, which is now the Coxiella burnetii. The Coxiella burnetii is the pasteurization indicator temperature, and destruction temperature for alkaline phosphatase is slightly higher than the temperature which is required to destroy the Coxiella burnetii, which is the indicator organism for the pasteurization. Now the enzyme is destroyed at pasteurization temperature, and this property of the enzyme has been utilized to test whether the milk has been properly pasteurized or not. And in this test we are making use of a substrate which is known as p-nitrophenyl disodium orthophosphate. This is the substrate on which the alkaline phosphatase acts, and then we also make use of buffer. That is sodium carbonate buffer we will use here. Then yellow color is produced if the milk is raw or not properly pasteurized. So if alkaline phosphatase is present after pasteurization, that will convert this p-nitrophenyl disodium orthophosphate to para-nitrophenol, which is yellow in color, and indicating that the milk has not been properly pasteurized or has been contaminated with the raw milk. In case milk is properly pasteurized, no change in the mixture will take place, hence no color will be produced. Obviously if ALP won't be there, then it won't react on the substrate, and then yellow color production won't be there. So this is the reaction. So this is the enzyme para-nitrophenyl phosphate, and then alkaline phosphatase act on it, and then we have the nitrophenol, which is yellow in color. And if it is not present in the pasteurization had been taken place properly, then the yellow color won't be produced, your para-nitrophenol won't be produced. Okay, now if we want to measure the ALP activity in milk, there are three methods. First, the rapid test, that can be done by naked eye also, or by Lowy bond comparator, that is routinely being used in dairy industry or in labs. And second method is spectrophotometric method, which is a routine method. And another spectrophotometric method is there, which is reference method and given by Scarer. So Scarer rapid phosphatase test it is called as Scarer rapid phosphatase test. So we will discuss about all these three tests in the coming slides. Now first let us see the rapid test for the measurement of ALP inactivity, that is routine test which is being done for alkaline phosphatase in the labs. So we require apparatus like water bath is required, low band comparator is required, test tubes are required, and then buffer which is used is sodium carbonate bicarbonate buffer. So preparing it, you dissolve 3.5 gram of anhydrous sodium carbonate and 1.5 gram of sodium bicarbonate in 1 liter of distilled water. So 3.5 gram of anhydrous sodium carbonate and 1.5 gram of sodium bicarbonate in 1 liter of distilled water. Then buffer substrate, you will use dissolve 0.15 gram of disodium paranitrophenyl phosphate in 1 liter of sodium carbonate bicarbonate buffer. The buffer which you have prepared, you will dissolve that is 0.15 gram of disodium paranitrophenyl phosphate in it. Okay, so now these are the steps which are involved to determine the activity in milk. So first we have to take 10 ml 5 ml of quantities of buffer substrate solution into the test tubes. Okay, so you will take at least 2 test tubes. One will be for the control, where we will make use of boiled milk, in which definitely alkaline phosphatase won't be there, and another milk which for which we want to check whether the pasteurization has been taken place or not in the another test tube. So take 5 ml of quantity of buffer substrate solution into test tubes, then keep at 37 degrees Celsius in a water bath. Then you add 1 ml of milk to be tested. Mix the contents in the tube. Prepare in the same way blank also from the boiled milk of the same type as under the test. Incubate all the tubes at 37 degrees Celsius in water bath, and after 30 minutes we will observe yellow color, and yellow color is read in level bond comparator that I will show you in the coming slide. All purpose comparator on a Rizogerin stand fitted with disc calibrated in microgram of paranitrophenol. Okay, this we will see in the coming slide. Then again you return it back after taking the reading using low band comparator. You return it back to the water bath and take a second reading after 90 minutes also. Now blank is placed after 30 minutes and 90 minutes, the 2 times reading to be taken, and blank is always kept to the left of the stand and sample on the right. Readings are taken by looking down onto the 2 apertures, and there will be 2 apertures with the comparator facing a good source of north daylight. So it is necessary that comparator will face a good source of north daylight. Now disc is revolved until the sample is matched. So sample color should be matched with the discs which are there in the low bond comparator. Readings falling between the 2 standards are recorded in nearest. If the readings are falling between the 2 standards, then you take the nearest one of the 2. Okay, so this is how low bond comparator 2000 plus looks like, and we have 2 test tubes here. You keep the test tube at their places, and then this is placed inside it, and then we will rotate the disc until the color matches with that of the sample color. So here on the left hand side we will take here on the left side we take the boiled milk, and on the right hand side we will take the sample for which we are checking the efficiency of pasteurization. Okay, now here disc will be inserted in this, and then the color of the sample. So here the whatever color will be there, that will be matched with the disc which we are rotating here, and once the color is matched, we will have a reading on the scale. So here this is the scale, and then we will have a reading on the scale, and then we'll interpret whether the pasteurization has been taken place or not. So let us see in the coming slide. So observation will be like this. So this reading after 30 minutes, if it is 0 or traces, then it means that it is properly pasteurized, and after 90 minutes it should not be more than 10, so indicating that it is properly pasteurized. Now if this reading is after 30 minutes is 6, then indicate that the pasteurization that the pasteurization is doubtful, or the pasteurization has not taken place. And then after 90 minutes the reading is coming 10 to 18, then it is slightly under pasteurized. And if initially after 30 minutes the reading is 10 or more, then it is definitely under pasteurized, and after 90 minutes the reading is ranging between 18 to 14.2, then it is under pasteurized, and if it is greater than 40 then it is grossly under pasteurized. So it is not at all it is not pasteurized. So this way we interpret whether the pasteurization has taken place properly or not using low bond comparator. Now there is another method which is spectrophotometric method, routine method. Let us see the procedure for it also. So you take 3.5 ml of carbonate bicarbonate buffer in a test tube and add 5 ml of buffer substrate, which is nothing but the paranitrophenyl and dihydro that is disodium phenyl phosphate, and keep the tube in water bath for 37 degrees Celsius at 34 minutes at 37 degrees Celsius. After the above pre-mentioned pre-combination, you have to add 0.5 ml of milk previously diluted 1:10 with the distilled water and mix the contents. The corresponding blank is carried out using the boiled milk inactivated enzyme. Now incubate the tubes for 30 minutes at 37 degrees Celsius. Then you add 1 ml of TCA to the tubes and mix the contents properly. Now purpose of TCA to precipitate the protein, and then filter the content of the test tube through filter number 1 filter paper, and then to 5 ml of filtrate in the test tube, add 0.5 ml of sodium hydroxide, because to have the alkaline environment, to have the optimum temperature for the alkaline phosphatase. Centrifuge if necessary. Measure the absorbance of the developed color in a spectrophotometer at 405 nanometer. Okay, so this is the step by step you are showing that if you want, we have to prepare standard curve. For preparing the standard curve, prepare a solution of different concentration of paranitrophenol 0 to 50 microgram. Like here we can see that 0 to 50 microgram solution. We are adding water first, that to have the total of 5 ml of the solution, and then you have to add 0.5 ml of 14% sodium hydroxide. Then concentration will be 0 to 50 microgram as shown on the slide. So 0 to 50 microgram of paranitol will be there in the solution to have the standard curve. So we will take the reading and then we will draw a calibration curve. So here the curve will be like this, and then for our sample, whatever reading is coming for our sample, say absorbance is coming, then accordingly we will be able to know how much microgram of paranitrophenol is there, and we will convert it into the enzyme unit. So calculation of unit of expression, express the result in terms of enzyme units. We will express the result in terms of enzyme units, and 1 unit of alkaline phosphatase is defined as the preparation liberating 1 microgram paranitrophenol in 30 minutes at 37 degrees Celsius. The concentration of paranitrophenol in the sample can be calculated using the standard curve, and all the sample giving reading excess of if the reading is excess of 10 microgram, then this indicates that the pasteurization is unsatisfactory, that pasteurization has not taken place properly. Now if the pasteurized milk giving reading less than 10 microgram of paranitrophenol, then it can be recorded as negative for alkaline phosphatase, or the alkaline phosphatase or the pasteurization has been taken place properly. Okay, now there is another third method is there, which is Scarer rapid phosphatase test, or the spectrophotometric method. This also is spectrophotometric method. In this, what is there is that we have substrate disodium phenyl phosphate to which alkaline phosphate acts, and then we have the phenol and the phosphate production. This phenol again react with the compound, the color producing compound, which is 2,6-dichloroquinone chloramide, CQC short form. It produces indophenol, which is blue in color. So this indophenol which is produced is blue in color, and the intensity of the blue color will be directly proportional to the activity of alkaline phosphatase or the concentration of alkaline phosphatase in milk. And so when milk containing alkaline phosphatase enzyme is mixed and incorporated with substrate disodium phenyl phosphate, the enzyme in raw milk cleaves a phosphate group from the disodium phenyl phosphate. So here you can see that it cleaves a phosphate group from the disodium phenyl phosphate and liberates phenol. Here we have seen that phenol. Now liberated phenol then reacts with the color producing compound 2,6-dichloroquinone chloramide to give a blue color. So we will have the blue color, which is indophenol, the compound which is produced in the fluid, which is having blue color. Now intensity of blue color depends on the enzyme activity present in the milk, and more is the enzyme, more phenol will be liberated, giving a deeper blue color. Okay, the intensity of the blue color can be observed visually or with a spectrophotometer, and that way we can have it in the qualitative form or the quantitative form, and the result of quantitative tests are expressed as micrograms of phenol per ml of milk. So quantitative test, we will express the results as microgram of phenol that is released per ml of milk. Amount of more than 1 microgram is indicative of improper pasteurization. So if phenol that is liberated is having greater than 1 microgram, then it is indicative of improper pasteurization. So calibration curve again we will do in this case also, and this is highly sensitive test, and it can detect contamination of pasteurized milk just by 0.1% of the raw milk. So it's highly sensitive. Even if 0.1% of raw milk is there in the pasteurized milk, then this will be able to detect the presence of alkaline phosphatase or the the efficiency of pasteurization. So how to do it? Simply, if we just to tell you, take 5 ml of the substrate in a test tube. We take 5 ml of disodium phenyl phosphate in a test tube, and to which you add 1 ml of milk. Okay, then you incubate the milk at 37 degrees Celsius, or the mixture at 37 degrees Celsius for 2 hours, and you will see that you can do it in either water bath or incubator 37 degrees Celsius for 2 hours. Then you will add the CQC, that is a 2,6-dichloroquinone chloramide you will add, and you will observe the presence of blue color. Okay? And then if blue color is not produced, then this indicates the milk is properly pasteurized, or there is no presence of alkaline phosphatase in the milk. So this is the third method, which is known as Scarer rapid test phosphatase method. Now there is another point which is to be shared with you, that is what is the reactivation of alkaline phosphatase. So sometimes it has been observed that the phosphatase negative milk, that is such as pasteurized or UST, immediately after processing become phosphatase positive on standing. So pasteurization has been taking place, and you keeping the milk for some time, and you will see that phosphatase is becoming positive. Now this reactivation can occur following heating and temperature as low as 84 degrees Celsius for milk and 74 degrees Celsius for cream. Then optimum storage temperature for reactivation is around 30 degrees Celsius, at which reactivation occurs after 6 hours. So if your optimum storage temperature for reactivation has been observed to at around 30 degrees Celsius, at which reactivation occurs after 6 hours. Now reactivation of alkaline phosphatase is considerable practical significance, since regulatory test for pasteurization assumes absence of ALP. So regulatory test, there are standards that ALP said test should be negative. ALP should be negative for the properly pasteurized milk. So that's why in that way it become considerable practical significance of reactivation of alkaline phosphatase. Now it has been observed that sulfhydryl group and magnesium ion are essential for reactivation. Now role of free sulfhydryl supplied by denatured whey proteins is considered to be the chelation of sulfhydryl groups which are free. They chelate the heavy metals, which in turn won't be binding to the enzyme sulfhydryl group, and thus preventing the renaturation. So otherwise, if the heavy metals would bind to the enzyme sulfhydryl group, their reactivation is prevented, and if free sulfhydryl group is there, then these will be binding to heavy metals, preventing their association with the sulfhydryl group, enzyme sulfhydryl group, and thereby preventing the renaturation. Now role of magnesium is seen as causing the conformation change in the denatured enzyme, causing necessary for renaturation. Now magnesium, how it causes the renaturation of alkaline phosphatase is by conformational change in the denatured enzyme, which is necessary for the renaturation. Now magnesium ion and zinc ion strongly promote the reactivation. So these magnesium ion and zinc ion, they strongly promote the reactivation, while the tin ion, copper ion, cobalt ion, EDTA are inhibitory, while iron has no effect on the reactivation. Okay, so this just a line that magnesium and zinc ion strongly promote the reactivation, but tin ion or copper ion, cobalt ion, EDTA are inhibitory, while iron ion has no effect on the reactivation of enzyme. So in this video we have learned all about the alkaline phosphatase test. Thank you.